So since 2 years, we are in a worldwide pandemic and still register high infection rates of SARS-CoV-2 on a daily basis. And also SARS-CoV-2 vaccination shows to reduce severe COVID-19 in the general population. There are still patients that do not benefit from COVID vaccination, and this includes patients under chronic immunosuppression. For example, after solid organ or hematopoietic stem cell transplantation, these patients receive high doses of immunosuppressants to prevent allograft rejection and by inhibiting the alloantigen immune response. However, also the beneficial immune response towards pathogens is inhibited, and that's why immunosuppressed patients often suffer from viral complications. And also COVID vaccination does fail to build up protective immunity in immunosuppressed patients, and therefore we need novel strategies to treat this patient cohort. And we propose SARS-CoV-2 specific antiviral T-cell therapy to treat immunosuppressed patients. However, the immunosuppressive environment limits the benefits of adoptive T-cell therapy in these patients, and therefore we generated SARS-CoV-2 specific T-cells that are rendered resistant to tacrolimus, which is an immunosuppressant that is widely used in the transplant setting. And therefore we isolated immune cells from peripheral blood of convalescent donors, and we stimulated these cells, SARS-CoV-2 specifically, isolated only the T cells that react to SARS-CoV-2, and in order to keep them active in the immunosuppressive environment, we rendered them resistant to tacrolimus by silencing the gene which encodes the FKBP12 adapted protein that is required for the immunosuppressive function of tacrolimus. And after the knockout, the SARS-CoV-2 specific tacrolimus-resistant T cells were further expanded until they are ready for infusion into the patient. And at the end of expansion, I performed the characterization of the T cell products on the single cell level using flow cytometry and also single cell RNA sequencing. And we could confirm the knockout on the DNA, RNA, and also protein level. And functional analysis has shown that the tacrolimus-resistant T-cell products are reactive and can secrete effector cytokines, also in the presence of tacrolimus when re-stimulated with SARS-CoV-2 specifically. And we also exposed these T-cell products to Cyclosporine A, which is an alternative immunosuppressant. And here we could abolish cytokine production. So this shows that Cyclosporine A could serve as an inherent safety switch in vivo, so in case any adverse events occur in the patient, we could shut down the T-cell products by applying Cyclosporine A to the patients. And also we could show that the tacrolimus-resistant T-cells are able to kill SARS-CoV-2 antigen-loaded target cells, also in the presence of tacrolimus. And also the transcriptomes by single-cell RNA sequencing were highly similar to an unmodified control T-cell product. So this really confirms the specificity of our knockout approach. And finally, we also checked for the cross-reactivity to common or occurring SARS-CoV-2 variants like Delta or Omicron, and also here we could show that the tacrolimus-resistant T cells are reactive to these SARS-CoV-2 variants. And therefore, like the GMP-compliant protocol to generate these tacrolimus-resistant T cells qualifies for translation into the clinic, and based on the promising results, we are currently preparing for a first-in-human clinical trial using vaccinated donor-derived SARS-CoV-2 specific T cells in the hematopoietic transplant setting, and we would like to confirm and validate like our approach in the clinic.